join us, then please do join us. And now I would request our Madam Chinmay Madam to start the session with introduction of the speakers and all. Thank you.
Thank you very much, Madam. Respected Dean Sir, respected the Vice Principal, Professor Madam, esteemed speakers, colleagues of our institution and other institutions, my dear students. A very good afternoon. On behalf of Department of Anatomy, SC Medical College, I welcome you to today's CME. Today we have two speakers for Platinum Jubilee CME. We are very lucky to have both of them with us now. Our first speaker is Professor Dr. Praful Kumar Chinara. He needs no introduction. He was the Dean and Principal of our institution and then continued as Professor and Head of the Department, Anatomy, Sam Hospital. He has a vast experience, teaching experience, and a number of publications to his credit. His area of interest is neuroanatomy, the most complicated part of our human body. Today he will be speaking on hippocampus and its mystery. Our second speaker is Dr. Priti Srivastava. She is an alumni of SC Medical College. She did her undergraduation in our institution from the year 1990 to 1996. She has 16 years of teaching experience in anatomy. She is now posted in North Delhi Municipal Corporation Medical College and Hindu Rao Hospital, Delhi as Associate Professor. She has many publications to her credit and she is the reviewer of many textbooks in anatomy. Today she will be speaking on the recent advances in development of cardiac septum. So I'll request now our first speaker, Professor P.K. Chinara, to start his presentation. Over to you, sir.
Esteemed Principal, Professor Dr. Sarvata, Vice Principal, Professor Saud, the faculties, my students, and other members of the medical faculty who have joined me this week. I am humbly honored and distinctly privileged to have this presentation in this prestigious Institute of Medical Science conducted by the Department of Anatomy, SC Medical College, Kotak. Now I have to present the hippocampus and its mystery. Hippocampus. There is always more thing in someone's hippocampus that you may not know. And there is always one more thing in your hippocampus that you may not know about it. Hippocampus is situated in the temporal lobe. The means it is a seahorse, Greek meaning seahorse. It resembles the parts of this seahorse. The left side is the hippocampus, dissected from the human brain, and that side is the seahorse. This is the dichromatic representation, and it is also known as the Cornoemonis or Amon's horn. Amon was an Egyptian god near whose temple the ammonia salt was prepared. When two hippocampi bend around, they form the form of a horns. Hence, it is also called ram's horn. This is the Greek god, and the two hippocampi bend forming ramshorn. These are the different types. If we, hippocampus is a complex structure, consists of, as you know, the cortex, the cerebral cortex, phylogenetically as the archaea and paleo and neocortex. Archaeal components are hippocampus proper, which consists of cornuamonis, dentate gyrus, subiculum, and parahippocampal structures are pre-subiculum, subiculum, entorhinal and perirenal cortex. When the brain is lateralized, two hippocampi located just over 1.5 inch over each ear. Deep 1.5 inch. It is area of cerebral cortex rolled into the inside. This is the situation of the cortex in different lateral, anterior, and ventral posterior view inside the brain. The dissected part of the hippocampus inside the brain. This is the ventricle, lateral ventricle, inferior heart. Hippocampus situated in the floor, forming the medial part of the floor of the inferior heart. Laterally, there is another eminence called collateral eminence. This hippocampus, about 5 centimeters long. The anterior head towards the tip of the anterior lobe is having paw shape. There are heightened paw shape called base hippocampus. Whereas the body and the tail goes towards the splenium of the corpus backwards. This is the another view at the sagittal section. This is the horizontal section of the hippocampus. And as I have told, divided into head, body, tail, and head. This is the base hippocampus, the head end, and I have described while it always forward. This is the base hippocampus. And the posterior end of the hippocampus continues at the fimbria and the fornix going forwards towards the anteriorly towards the mammillary body, hypothalamus and to septal area. Next. So from this hippocampus, one side fornix, other side fornix. Here the fibers goes to opposite hippocampus, forming the commissure of the fornix or the hippocampal commissure, and anteriorly in front of the anterior commissure, pre-commissural, post-commissural part, pre-commissural part to the septal area, post-commissural part to the hypothalamus and the tegmentum of the brain. Next. Same thing in anteriorly, there is presence of amygdala. Next. See how the parahippocampal gyrus is turned inwards, and it is separated from the medial occipital-temporal by the collateral sulcus. This collateral sulcus going deep, producing elevation called collateral eminence in the floor of the lateral ventricle. There medially there is the hippocampus. This hippocampus has 3 parts. It is S-shaped. The upper limb of the hippocampus, including dentate gyrus, middle is the subiculum, and the another is the parahippocampal gyrus. This is the different part diagrammatically, dentate gyrus and the hippocampus proper, subiculum, and entorhinal cortex. Next. Let us discuss about the dentate gyrus. The dentate gyrus, this is the dentate gyrus. Superiorly there is the cornuamonis. Also the laterally there is the inferior horn of the lateral ventricle. Inferiorly the subiculum. Medially, superomedially there is the fimbria, and inframedially the borders are free to form the dental structures, dental projections. That's why called dentate gyrus. The anterior end of the dentate gyrus is called tail of the dentate gyrus, which extends towards the uncus. This one is the uncus, limited by the rhinal circles. This uncus, the tail of the dentate gyrus, divides the uncus anterior part, the consonant gyrus, posterior part, intralimbic gyrus, and the posteriorly it continues as the gyrus fasciolaris or the splenial gyrus above the corpus callosum, and a layer of gray matter called inducium gyrus, which goes forwards towards the septal region. Next. This is the another view of the dentate gyrus, hippocampus, subiculum. Next. If you see the structure of the dentate gyrus, the cortex has the outer molecular, middle granular, inner polymorphic cell layer. So in the hippocampus and dentate gyrus, these three layers are there. In the middle layer, in the dentate gyrus, there are granular cells, and in the hippocampus, in the middle layer, there is the pyramidal cells. Otherwise, the outer molecular, inner polymorphic layer, same three layers. Next. See, here is the those layers. This is the granule cells in the granular layer. This is the molecular layer having the dendrites of the granule cells. The axon passed through the polymorphic layer, and there are other cells, interneurons. Next. The hippocampus, otherwise called as cornu-emonis proper, is divided into 4 parts. As I have shown in the previous diagram, the CA1 cornu-emonis, CH cornu-emonis 1, 2, 3, 4. 4 is not inversely accepted. C2, 3, and this 3, 4 towards the dentate gyrus. Similarly, as I have told, it has the 3 layers. The middle layer is the contains pyramidal cells, and these pyramidal cells have the axon going towards the polymeral cells, and superiorly there are the apical dendrites, and inferiorly are the polymorphic layer, there are the basal dendrites. This is the cell. The molecular layer again have 3 subdivisions: stratum moleculi, lacunosum, radiatum. This is molecular layer. And pyramidal stratum pyramidals contain the pyramidal cells, and the stratum aureus polymorphic cell layer, deep to which there is the alveolus. Alveolus is nothing but these axons of these pyramidal cells. They form a layer over the cornu-emonis, deep to the ependime of the ventricle, the white matter called alveolus. Next. Next is the subicular complex. The subiculum is divided into the subiculum, pre-subiculum, parasubiculum. Actually, after the cornu-emonis, the part immediate is the prosubiculum, then subiculum, then pre and parasubiculum. This contains pyramidal neurons that project to entorhinal cortex. Next. This one is the dentate gyrus, different layers. This is this region 4, 3, 2, 1 of the hippocampus, and the different layers. Towards the ventricle is the deeper layer, polymorphic layer, and here is the molecular layer, because it has take the contour shape. That's why the superficial layer appears to be deep, and the deep layer appears to be superficial. But this is the deep layer. Next. The entorhinal cortex, another part of the parahippocampal gyrus, the anterior part of the parahippocampal gyrus, entorhinal cortex confined laterally by the collateral surface, and the perirhinal cortex buried in the wall of the collateral. The entorhinal, perirhinal are the entorhinal complex. And the cortex is of six layers. We know the neocortex 6 layer, and the archicortex hippocampus 3 layer. The entorhinal cortex is of 8 layers. Here the second layer, pre-alpha layer, has the densely packed pyramidal cells. This is the hallmark of entorhinal cortex. And the 5th layer, lamina dissidens, is acellular layer. This is the distinctive feature of the entorhinal cortex. Next. Then the parahippocampal gyrus. So dentate gyrus, hippocampus, the subiculum, and the entorhinal cortex continuing the parahippocampal gyrus. Parahippocampal gyrus provides a path of communication between the hippocampus and all cortical association areas. Next. And just to complete the anatomy portion, the blood supply of the hippocampus is by posterior cerebral artery, its anterior middle posterior branch, and also reinforced by the anterior choroidal artery, truncal branch of and veins to the basal vein. Next. This is a intrinsic neuronal connection. We'll see the intrinsic. This is the dentate gyrus containing the granule cells. It's the axon, and these are the dendrites. And this one is the pyramidal cell. Pyramidal cell, it's axon, and there are two basal dendrites and apical dendrite. The hippocampus dendrites in this superficial layer, molecular layer, make synapse with the fibers coming from the renal cortex, that the perforant fibers, perforant pathway. And its axon goes to the dendrite of the pyramidal cells, as mossy fibers, mossy fibers. Then the pyramidal cells, its axon of the pyramidal cells forms a layer, the alveolus, deep to the ependyma, then continues superomedially to condense at the fimbria, and this fimbria will continue as a fornix toward the splenium. Whereas the basal dendrites, they synapse with the two structures, one commissural fiber coming from the opposite hippocampus from the identical areas, and one recurrent collateral from neighboring pyramidal cell. But the apical dendrite in the molecular layer makes synapse with perforant pathway from the atrial cortex, fibers from the mossy fibers from the dentate gyrus, fibers from the supramammalar region of the hippocampus, the hypothalamus, and commissural fibers from non-identical areas of the opposite hippocampus, and recurrent fibers from the recurrent collaterals from the neighboring pyramidal cells of the cornuamus. This is the neurons. Next. So just the perforant path perforate the subiculum, and this is unidirectional. Next. These are the dentate cells, dentate gyrus, cornuamus, cornucopia C. These are all unidirectional, and these are the connections. Next. This is another same thing, but in another way. This is the basic circuit that I have told from the perforant path from entorhinal cortex to dentate gyrus, CA3, CA1, dentate gyrus mossy fibers to CA3, and suffers collaterals from CA3 to CA1 suffers collaterals. Next. This is the same circuit diagram from entorhinal cortex, dentate gyrus, CA3 up to subiculum. Next. This is more larger part, but only the frontal cortex, cortical areas. Next. The inputs to the hippocampus comes from the cingulate, temporal, orbital, olfactory cortices, and from the anterior aspect amygdala. So from all cortical portions, ultimately the input is coming to the hippocampus through the parietal gyrus, entorhinal cortex, perforant pathway, alveolar pathway. And the output, main output is the fornix. From the hippocampus, the fornix, fimbria, fornix, then body of the fornix going forward to septal region, hypothalamus, there to the thalamus, then cingulate gyrus, and others efferents are going from the hippocampus to entorhinal cortex, and so this is the output pathway. Next. Here we may discuss the Papez circuit. Papez circuit basically starts from the hippocampus. See, this is the hippocampus. From the hippocampus through the fimbria through the fimbria come to the hypothalamus, mammillary body. Then goes to the anterior nucleus of thalamus. Anterior nucleus of thalamus. From there goes to the cingulate gyrus, and from the cingulate gyrus it's come again back to the hippocampus. This is the Papez circuit, original Papez circuit. Next, and this thick line, these thick lines are the original circuit, and thin lines are later added in the Papez circuit. Next. So hippocampus is very well connected to other brain areas, other up to frontal, orbital, cingulate, piniform, perirenal, striatum, amygdala, septum, mammillary body, thalamus, hypothalamus. Next. So that is overall about the anatomy of the hippocampus. Let us see what is the function of the when the function of the hippocampus comes. What did the hippocampus say at its graduation level? Gives thanks to the memory. Means memory is related to the hippocampus. Some theories of hippocampal function are that it is like just an index which bind together elements of an experience but does not store experience itself. Latter, it is distributed in a throughout the brain, not confined to a particular part, throughout the brain. Next, two most influential theories of the hippocampus are related to space and memory. The spatial hypothesis suggested that hippocampus was a sort of device used for the mapping layout of the environment, and virtual navigation studies in humans suggested a strong association between hippocampus and spatial navigation. We come to that one. The memory hypothesis originated in 1957. The hippocampal removal resulted in loss of ability to form new memories, particularly fact and event related, declarative memories. Exact process by which the hippocampus supports memory subject to much debate. So when the memory comes, first the automatically the memory comes towards the Henry Molaison, just shortly called as HN, who suffered from epilepsy and generalized epilepsy, say 2 to 3 generalized per week and daily some minor seizures. And in his late 20s, he underwent operation in 1953. Both the hippocampi, bilateral hippocampi, was removed by the temporal lobectomy. Lobectomy means whether a part of major part or total lobe is removed. And this is after the surgery. After the surgery. And next. And he lived in a care institute at Connecticut, Windsor Law, Connecticut. And this study on his memory was done from 1957 up to 2008 till his death. Thereafter, the brain is collected and also next. Let's slice as this is the brain of Henry Molaison removed, hippocampus removed, bilateral removal, and this is normal brain. Next. So the why I am taking about the Henry Molaison, because after the surgery, when he recovered his consciousness, he suffered from amnesia, both retrograde and anterograde amnesia. Retrograde, what he has done previous to the surgery, just immediate, say 2 to 3 days before the service, he can remember. And after the surgery, what he does, he can't remember. That's anterograde and retrograde amnesia. But he can remember the address of his home, the long past events. Let us see the memory. Hippocampus itself plays important role in human beings to process everything. Memory is divided broadly into explicit memory, implicit memory. Explicit is declarative memory, which is again further divided into episodic and semantic and procedural memory. The episodic memory for specific events. Suppose you remember the name of your class teacher, wedding date of your relation. These are specific events, that is episodic memory. And semantic memory, recollection of facts, say difference between a cat and a dog. And say to produce few lines of the play, semantic memory. That is declarative or explicit. Implicit memory is automatic memory, unconscious memory. This is through practice, acquired through practice. Say you have learned how to ride a cycle, even after 50 years, even you have don't touch the cycle for last 40 years, you can write it well without remembering what are these different processes. So this is the unconscious memory. The phases of memory are the registration, storage, and retrieval. Memory consolidation, memory long-term LTP is the mechanism for storage of the memory, long-term storage, and bilateral damage, impairment of short-term memory to the long-term memory. The other functions, decision making, the hippocampal damage, unable to live independently, hold the full-time employment. Spatial navigation, when somebody is remembering the routes or the place where the school is placed, the place cells are there. At these places, when you enter the particular element, the place cells are activated, and you remember. Suppose light goes out, and you can locate where the switch is because it is there in your mind. So even without knowing, in the darkness, you can go and reach the switch. And spatial navigation, a driver know the routes. It has been seen that the drivers who have driven 20 years in the London streets, their posterior part of hippocampus has more thick gray matter, because to remember the routes, charts, the positions, that is spatial navigation. Then imagination, creativity, hippocampus involved in imagination and creativity. There are hippocampus and learning through eye blinking condition, and damage to hippocampus produce maladaptive behavior, and it is also related to make appropriate character judgment by contextual information, and has social the quality of memory contribute to empathic response and social discourse and languages. In man, it influences endocrine, visceral functions, and emotional states through connection with hypothalamus, hence considered as the seat of the soul. And in 2016, they published that the exercise regenerate the new cells in the hippocampus. Though the neurons do not undergo mitosis, still there are some areas, and in old age, exercise may regain the strength of the memory by regenerating the new cells. That's why exercise is very much essential, and morning walk is. And in 2017, in Hong Kong, they published a paper that low frequency activities in the hippocampus influence the functional activity of a brain, leading to increased sensory response such as vision, hearing, and touch. Hence, in this sense, it could be described as heart of the brain. And the disease, the hippocampus is a sensitive part of the brain. Range of conditions can adversely affect and impair the hippocampus ability to do its job. In Alzheimer's, epilepsy, depression, the size, there is a small size hippocampus. The size of the hippocampus, smaller and smaller, determines the progress of the Alzheimer's. And in epilepsy, there is in 50 to 75 people, there is damage to the hippocampus. In depression, 10 to 20% size shrink of the hippocampus. Stress also causes damage to the hippocampus. And so main thing is the memory. Memory, as I have told, there are two types of memory. Say you are coming to your home and suddenly you are ordered that bring 5 things from the store. So with the bike, you will remember 5 things, you will go to the store. With the bike, you are going, not remembering. That is the implicit memory, implicit memory. Where the 5 things, what you will bring, you are counting in your mind. That is the declarative memory. And this is the prosthesis of the hippocampus. Human prosthesis developed in 2013. Thank you.
Thank you, sir, for your nice presentation of the most complicated part of the human brain. Our question answer session will be at the end. Now I request Madam Kirti Madam to please start your presentation.
A very good afternoon to all of you, respected Vice Principal Madam Geeta Sahu, Professor Dr. Geeta Sahu Madam, respected Professor Dr. Chinmay Madam, and all the respected faculty of the Department of SCB Medical College and all the faculty who have joined us and are linked to us with the in this free platinum Jubilee CME. I am an SCBN and I joined in 1990, approximately 31 years back. But today I'm feeling very proud having been invited to be a part of this Luckily Platinum Jubilee celebration. And I extend my heartiest congratulations to all who are associated with SCB Medical College. It's an honor of highest order, I feel, that I'm able to speak to my teachers from a dais, and my teachers are willing to listen to me like students. With pride in my heart, I am here to speak on the recent advances in development of cardiac sector. Why did I choose the recent advances in cardiac sector? Is because the incidence of congenital heart diseases is pretty high, approximately 0.8% to 1.2% of live births globally. But the septal defects are the major subtypes of the congenital heart diseases. And with the color coding that you are able to see from the incidences, you can see that the Africa and Asia are very highly affected. And it is probably because of poor nutrition and infection during pregnancy, which affects the fetus and leads to congenital heart diseases. And you can see the highest incidences being on in Somalia, in Burundi, and in Central Africa. Now, before we proceed further, I would like to describe the normal development of heart. From day 16 onwards, there is the migration of heart progenitor cells from the primitive streak, which you can see here. These are the primitive streak. The cells are migrating and they are going cranial to the neural fold and forming a crescent-shaped primary heartbeat. Then the lateral body folding occurs, as a result of which, and because of vasculogenesis, which is induced by the endoderm, heart tube formation occurs approximately day 20 and 21. The lateral folds of the fetus occur, lateral fold of the body, and the heart tubes come to the lie close in the midline. Then by day 22, the heart begins to beat. Day 23, heart begins to loop, and day 28, the looping of the heart is complete. And here you can see that some amount of septation has also started to develop. Then between day 33 to 35, you can see that the endocardial cushion tissue formation occurs, and outflow tracts endocardial cushion begin first. Later, the AV cushion, endocardial cushions begin to develop. At the same time, approximately by day 63, one can see that outflow tract septation occurs, and also the ventricular septation has, the muscular part of the ventricular septum has almost been completed. Now coming to a little bit about the gene expression in the heart tube. In this diagram, you can see that the blue color is the neural tube, the yellow color tube is the gut or the foregut, and the red one is the heart tube. And here you can see that the endoderm of the foregut secretes bone morphogenetic protein, and from the neural tube comes the Wnt pathway, where the Wnt expression. But inhibition of Wnt expression combined with secretion of bone morphogenetic protein induces expression of NKX2.5 from the heart tube. Now, this NKX2.5 is considered to be the master gene of development of heart. Slightly talking about how the development of heart and talking about what exactly is cardiac jelly. Much talked about topic. Now here you can see that the heart tube is made up of a myocardium, which is the outermost mantle. Then there is an acellular, extracellular matrix, which is called as cardiac jelly, and there is an endocardial tube, which is lying inside. The cardiac jelly, in order to be called as or termed as cardiac jelly, must occupy the myoendocardial space. That means between the myocardium and the endocardium, the space that is occupied should be cardiac jelly, should be called as cardiac jelly, provided it is acellular. It should be free of cells. And this cardiac jelly is present only through Carnegie stages 10 and 11. That means the cardiac jelly will disappear. But we know that cardiac jelly is present or persisting at certain areas. Now, what are the areas? The cardiac jelly persists at AV junction and the outflow tract region. So it causes a swelling. These areas are devoid of cardiac jelly, whereas these areas have accumulated the cardiac jelly. And here, these swellings are called as endocardial cushions or the AV cushions. And where the endocardium undergoes an epithelial to mesenchymal transformation, which we now henceforth call as endo-EMT, and they give rise to some cushion cells, which are endocardially derived mesenchymal cells, and then it is no longer called as cardiac jelly because now it contains cells. These fuse like this to make an AV septum. It also gets a contribution from the neural crest cells, contributing to AV cushion formation. Now, failure of migration of neural crest cells will obviously cause congenital heart disease. Now, coming to the genesis of AV septal defects. The accepted paradigm till now is that if there is any perturbation in the development or fusion of the endocardial AV cushions, they are responsible for atrioventricular septal defects, and they are generally called as endocardial cushion defects. The recent studies have linked abnormal development of the dorsal mesenchymal protrusion. This is the in thing which is there. That is, the dorsal mesenchymal protrusion is what affects atrioventricular septal defect. So, what is this dorsal mesenchymal protrusion? Dorsal mesenchymal protrusion is a part of the AV mesenchymal complex. AV mesenchymal complex has three components. The ones are the mesenchymal AV cushion, second one is mesenchymal cap on the primary atrial septum, and the third one is the dorsal mesenchymal protrusion. So here in this diagram, I've tried to show all the three. They are almost at this area, that is the atrioventricular area. We will talk about it in the detail in the subsequent slides. So basically, we are going to concentrate on dorsal mesenchymal protrusion in the end. Now, what happens at the end of the first trimester? The cardiac jelly which was accumulated has converted into cushions in the cushion cells because of endo-EMT. The superior and inferior, there are 4 cushions, out of which the major ones, which are superior and inferior, they meet in the midline to create an AV septum, and hence after that the common AV canal, this common AV canal is divided into a right AV canal and a left AV canal. And here one can see that 2 lateral endocardial cushions are projecting. Now, out of these, the right lateral cushion gives rise to the parietal leaflet of the tricuspid valve, whereas the left one gives rise to the posterior leaflet of the mitral valve. Any EMT defect here, that is endo-EMT, where the cushion cell formation occurs, that also affects non-migration of neural crest cells, and this will result in defective AV cushion formation. Now, formation of atrial septum. Commonly, we know that the septum primum develops, then septum secundum develops, but here we have to talk about another element, and that is a dorsal mesenchymal cap. So this is being highlighted with a red star. Dorsal mesenchymal cap is nothing but derived from endocardium. It is forming the leading edge. It is completely covering the leading edge of septum primum, and it is endocardially derived, whereas septum primum is myocardially derived. Hyperglycemia and hypoxia during pregnancy will inhibit again this EMT formation, which will result in congenital heart diseases. So here we have to keep in mind that out of the AV mesenchymal complex, we talked about the AV cushions, and these are the dorsal mesenchymal cap, which is there on the primary atrial septum. Now we come to the DMT, which we are talking about. This is also called as spina vestibuli or the vestibular spine. Now the mesenchyme of the dorsal mesenchymal protrusion, I'll show you. This is what is the dorsal mesenchymal protrusion, and this is dorsal mesenchymal cap. So dorsal mesenchymal cap, sorry, the dorsal mesenchymal cap with the dorsal mesenchymal protrusion, and this is the AV cushion. Together, they make the AV mesenchymal complex. Now, dorsal mesenchymal protrusion, what is so different about it? This is also a mesenchyme, but it is not derived from the endocardium. It is derived from the second heartbeat. Now, what is second heartbeat? All of us know about the primary heartbeat, which is also called as the first heartbeat, which is in the form of a crescent shape. Now, medial and inferior to it lies the second heartbeat. The second heartbeat is the source, and the endocardial cells, the mesenchymal cells derived from the second heartbeat are the core of the dorsal mesenchymal protrusion. So any protrusion, the protrusion from into the common atrial center from the dorsal aspect containing mesenchymal cells is dorsal mesenchymal protrusion. In this, you will be able to see that in this diagram, this is the foregut which has been highlighted. This is the pit where the pulmonary vein develops. In between lies the dorsal mesocardium. Now, these brown nuclei are nothing but which are expressing ISL1 expression, and they are trying to locate in the second heartbeat. So, this is the area of the second heartbeat as well. Now here the mesenchyme endo-EMT occurs, and the second heartbeat will give rise to a dorsal mesenchymal protrusion like this here, which is seen here. Now in this picture, the red ones are nothing but the myocardial fibers, and this is the dorsal mesenchymal protrusion. This is the second heartbeat. This is the foregut. This is the primitive pulmonary vein. So you can see that dorsal mesenchymal protrusion is actually to the right of the pulmonary vein, and hence it is pushing the pulmonary vein to the left. As a result of this, the pulmonary vein will incorporate into the left atrium. Recap of atrial septation, atrial septa formation. This is primary or the primum atrial septum with a mesenchymal cap. This is primary foramen ovale. Then primum foramen, sorry. These are the endocardial cushions which are mesenchymal in origin. Now, with perforation above, we have the primary atrial septum with the mesenchymal cap fusing onto the dorsal mesenchymal protrusion and AV cushion. Now, here you can see AV mesenchymal complex forming. That is the mesenchymal cap, the dorsal mesenchymal protrusion, and the AV cushion. And therefore, with the myocardial element of the primary atrium and the secundum atrium, atrial septa, the atrial septal formation occurs. In the end, the dorsal mesenchymal protrusion is myocardialized, and therefore it forms a muscular rim of the atrial septa, which is seen from the right side, and it is called as muscular rim. The molecular regulation of dorsal mesenchymal protrusion. So here you can see that bone morphogenetic protein, this one, is actually affecting, and it is in absence of BMP4, we find that there is lack of development of dorsal mesenchymal protein. The second one is the Wnt beta-catenin pathway, and this also will is essential for the development of second heartbeat and the dorsal mesenchymal protrusion. The third one is sonic hedgehog. In sonic hedgehog pathway, if there is deficient conditional deletion, then it leads to a single outflow tract. That is called as persistent truncus arteriosus. Then we come to the PDGF or the platelet-derived growth factors and its receptors here. If we have, in experimental conditions, the receptor knockout mice will show hypoplasia of dorsal mesenchymal protrusion. This is TBX5, and TBX5, if there is haploinsufficiency, it results in Holt or Ram syndrome, which leads to limb and cardiac defects. Now, the primary cilia has intraflagellar transport protein, which is responsible for, which acts via the sonic hedgehog pathway, and ciliary dysfunction causes various types of AVSDs or the atrioventricular septal defects. The last but not the least is heterotaxy, which is also leads to situs inversus, dextrocardia, ventricular inversion, and these are all because of the defect in ciliary motility. So this is the primary cilia, and this was a model of a posterior second heart field DNP precursor cell. Now, in between, before the ventricular septation occurs, there is realignment of the chambers. So what happens in realignment? Basically, there is a common atria here. The common atria has an outflow into the left ventricle, whereas the right ventricle has a common outflow. So inflow is into the left ventricle. From left ventricle through the interventricular foramen, the blood comes to right ventricle and is going out via the outflow tract. So the right ventricle is actually not having an outflow tract, and left ventricle is not having an outflow tract, whereas right ventricle is not having an inflow tract. So this portion, which is given here, this portion which is called as the primary muscular fold or the bulboventricular ridge, undergoes a lot of remodeling, and there is myocardialization occurring here. There is ballooning of the chambers. There is formation of the muscular part of the interventricular septa, and this leads to realignment. Now, with the formation of AV septa, the common AV canal is split into a left AV orifice and a right AV canal. Into a right AV canal and a left AV canal. So here now in the end, both the ventricles have an access to the outflow tract, and both the atria have an access to the inflow tract. Now, development of the interventricular septum. Commonly, we know that they develop from a membranous part and a muscular part, but we do not. But lately, it has been found that the membranous septum in the first region, in the first picture, is made up of an atrioventricular component which has a contribution from the aortic valve, whereas an interventricular component of the membranous part has a tricuspid valve contribution. Whereas in the muscular part, now in this diagram, the right ventricular wall has been removed, and we are seeing the septum from the right ventricle. And you can see that the muscular part has three parts: the inlet septum, the outlet septum, and the trabecular septum. So trabecular is basically the main anterior aspect, which is called as the main muscular part. The inlet septum is contributed by the inferior AV cushion, and the outlet septum is contributed by the outflow tract cushions. Now, here what happens, the phonotruncal septum is now developing, and this is a traditional diagram which we explained, in which the aorta is lying anterior and the pulmonary trunk is lying posterior. And these are the two phonotruncal cushions which are meeting in the midline, and as a result of which, first the aorta lies anterior. Aorta lies anterior and the pulmonary trunk lies posterior. Then slightly above, there is the anteroposterior. You can see the yellow portion is going behind. The anteroposterior twisting the septa lies like this, and then the aorta comes to the right, Whereas, sorry, the pulmonary trunk comes to the right and the aorta comes to the left. Later, aorta comes to the anterior aspect and pulmonary trunk goes to the posterior aspect. Now, this is okay, but in this, there is also contribution of the AV atrioventricular septum contribution here, as you can see, and here there is a mesenchymal wedge tissue which will be incorporated in between these two and splitting the two tracts, making it formation of aorta and pulmonary trunk. Neural crest cell contribution throughout, we've been seeing that the neural crest cell contributes to the outflow tract septation. Any defect in neural crest cell migration leads to persistent truncus arteriosus, tricuspid stenosis, ventricular septal defects, transposition of the great arteries, double outlet right ventricle, and tetralogy of Fallot. Defect of both the outflow tract and pharyngeal arches includes a Chart syndrome, a fetal alcohol syndrome, a DGeorge or a cardiofacial syndrome, which includes a 22 deletion syndrome. PBX1 leads to persistent truncus arteriosus. Neural crest cells also regulate the expression of several other genes within the second heartbeat, which includes glucosid, DLX2 and DLX3, as well as HANG2. These are the types of ASDs and BSDs. Depending on the components, here you can see this is ASD type 1, which is also called as ostium premium defect. This is BSD type ASD type 2, which is ostium secundum type of defect. These are the superior and inferior vena cava contribution, and these are called as sinus venosus defects if they do not incorporate properly. And here lies the coronary sinus defect. Now in BSD, we all have 4 parts. So one is conoventricular BSD, then we have the perimembranous BSD, we have the inlet BSD, and we have the muscular BSD. Take home message is the classical view of the septum formation. So classical view was that the atria is formed by septum primum and septum secundum. Then in ventricular septal formation, the classical view is that they are formed from the membranous part and the muscular part. But the recent studies told that there is a secondary cardiogenic area which gives mesenchymal cells to the dorsal mesenchymal protrusion. So contribution of BMP is there in the atrial septa. There is also other mesenchymal contributions like the mesenchymal cap, the AV cushions which have the mesenchyme, which is due to the endocardial epithelial mesenchymal transformation, and there is also contribution from the neural crest cells. We cannot rule out the contribution of genes and the clinical syndromes associated with it, like NKX 2.5 is the master gene of development of heart, and TBX5 and TBX1 lead to Voltoram and persistent truncus arteriosus kind of syndromes. Thank you. No presentation, no work is complete without the entire contribution from the department. This is my department of anatomy. This is my HOD, Dr. Tara Shankar Roy, myself, Dr. Preeti, my assistant professor, Dr. Swati, my 2 senior residents, Dr. Bhaskar and Dr. Shivani, Mr. Muniruddin and Mrs. Usma are our lab assistants. Ms. Daljit and Mrs. Meenakshi are our lab attendants. Thank you.
Thank you, madam, for updating our knowledge on recent advances in development of cardiac septum. Now, there's a time for answering some questions which are asked. Question. We know we have a very
But we find it difficult to understand this
And
subject. That you find help us.
Could you please help us
Give us some tips how to master.
this subject? How to master this subject? Answer, sir, answer.
You learn the subject Master cannot master it. What about the students? But you have to repeatedly read it, and the basic fundamental understanding is the main thing. Fundamental understanding. Just like the gross anatomy. If you have dissected the structure, you know it. If you have read it, you do not know it. Similarly, neuroanatomy, you have to understand the basic thing. And it is just like electrical circuit. You have gone to some part and somewhere you are blocked, the entire thing is gone from your mind.
Next, I would wish. It's
Who can
interesting.
see your assets?
It is interesting to know about the gentleman itself and his memory loss. Could you tell us something more about background history of HL?
Yes. Actually, to preserve the anonymity of the person while living, we tell it as HL. Otherwise, it is venerimalization. Who suffered epilepsy for 11 years. And in 1953, at the age of 27, he got operated removal of both the hippocampus. And after the surgery, as I have told, he developed amnesia, both retrograde and anterograde. That's why previously for any fits, epilepsy, they are removing the hippocampus, thinking that it is the focus for the seizures. So when they removed, this operation was an unqualified success. Yes, he was relieved from the seizure, but he developed amnesia. He lived a life of present only, but he has done yesterday he can't remember. Yesterday, have met him, he can't remember. That's why the researchers were interested to study about the memory with him while he was living at the care hospital in Inshallah, and after his death, even his brain was preserved, sizes were done, and it was studied which part of the brain is seizure and how much hippocampus, what are the changes in the hippocampus. As you know, already they are removed, but the brain changes that started after his death. While he was living, the changes after the surgery was studied with keeping him in the hospital.
Any other question?
Sir, I have a question. I'm Bipin.
Sir, sir.
Okay.
Sir,
The question is to Professor Chinara.
Okay.
In our books also, there is no detailed mention of the other pyramidal cells. You have said C1, 1, 2, 3, 4, CA1, 2, 3, 4. But no mention about the functioning or connections of those pyramidal cells are mentioned. Only C3 is connected to C1 by collaterals, what you have mentioned as Kaffir's collaterals.
Collateral.
Our books also silent about those things. Do you have any information about CA2 and CA4?
No, that I have told. They have divided histological division, anatomical division. Anatomical is regionally divided. C4, 3, 2, 1 was the subiculum. C4 is not that I have told. C4 is not universally accepted. So
No, C4 is not accepted, but C2 is accepted. CA2.
C2 is accepted. The fibers, the fibers from the granule cells, the axon of the granule cells comes as ossified to C3. C3. Actually, this C1, 2, 3, 1 contains 90% of pyramidal cells. 2 contains compact pyramidal cells, and 3 contains large pyramidal cells.
So C3 is connected to C1.
C3 connected to C1, but the literature is giving C3 and C2 connected to C1. Which is
C2 and C3.
C2 and C3.
Because our books don't write, even advanced neurophysiology books don't write about the function of C2 cells.
C2 is getting fibers, perforant path. is coming to dental gyros C3, C2, C1, everywhere. So the literature is mentioning C3 and C2. First, the mossy fibers is coming to C3, and from C3 and C2, the scapula collaterals are going to C1.
Acha. Now our book says C3 to C1 through scapula collaterals.
So,
No, not silent on C2.
so majority they don't accept C4. C2 is in ambiguity between anatomy and physiology.
And more so, we talk about hippocampal formation, that is dentate gyrus, hippocampus, and subiculum.
Grossly, hippocampus means we mean only hippocampus, but hippocampal components are dentate gyrus, hippocampus, subiculum.
Subiculum.
Subiculum is the joining area, it is transitional area, it has 5 layers. Neocortex, sectoral cortex, 8 layers. It has 5 layers, and archicortex 3 layers.
And your diagrams were very beautiful. Need not explain, it is self-understood, sir.
No, just I have.
Thank you for the beautiful diagrams.
Thank you. Copy and paste.
Thank you.
Any other question? So we'll end here and just give the vote of thanks. I'd like to extend my heartfelt gratitude to our respected Dean for initiating this academic series event as a part of Platinum Jubilee celebration. We all have attended CMEs conducted by our esteemed colleagues, which has enriched our knowledge and developed a sense of curiosity to learn more. All of this would have been impossible without Dean's hard work and encouragement. I would also like to thank our invited speakers, Dr. Professor Chinara and Dr. Preeti, for dedicating their valuable time and sharing their expertise with us. Last but not the least, I also thank my dear colleagues for attending this CME session and helping us in making it a successful event. Thank you.